the kind introduction, and Devaka and Gaurav for the invitation to the webinar. As I said, I'm really excited and honored to be sharing some of the work with members at Center for Computational Biology at IIIT Delhi, whose work from different groups coming out from the center I've been following more actively, and have interacted with to varying degrees with different members, Vibhor, Sriram, Debarka, Dr. Pratesh, Ganesh, and of course, Professor Raghav. So it's an honor for me to present some of the ongoing work in our group here, and get your feedback, as well as also explore potential synergies that we can acquire by working together in the broad area of systems biology and precision medicine when it comes to cancer metastasis. I hope my screen is visible.
Yes, it's visible. Yeah, I can confirm.
So the questions that we look at in our group that we try to answer relate to cancer metastasis and drug resistance in the context of cancer. And one of the themes that has been coming up repeatedly in the literature is that treating metastasis as well as the resistance that is developed against all the ongoing drugs, one of the major challenges, the clinical bottlenecks, is the idea of heterogeneity, that every tumor is different and every patient is different. Even within a tumor, every cell is different. So that's the overall idea of intra-tumor heterogeneity and intra-tumor heterogeneity, which every tumor can be made of more than one genetic clone, so they have different properties. Some cells may be more or less sensitive to the drug that is being given to the patient. So, of course, there has been huge progress in the methodologies that have been developed since to be able to characterize this heterogeneity and then see, can we parse through this heterogeneity, this variability, and then try to arrive at some more targeted therapies, which are referred to as precision medicine, largely speaking. So drug-drug combination, sequential therapies, and so on, and what can eventually have the impact on the clinic when it comes from bench to bedside. So in the context of heterogeneity, major focus of the work that has been coming around is on the idea of genetic heterogeneity, as I alluded to in the previous slide. In the sense that when a cancer initiates in a single cell or a group of cells, there is a set of genetic mutations, which is what sets it apart from a normal behaving cell. So this set of mutations is somehow able to either make the cancer cell insensitive to anti-growth signals or make it insensitive to the cell cycle arrest and various other features that are considered as hallmarks of cancer. And as cancer proceeds in a given patient, there are different bottlenecks that it faces because the environment in which cancer is growing is also a dynamic one, and which leads to a selection of certain properties which are finally imprinted in the DNA in terms of genetic mutations and other genomic rearrangements that may follow. So with information that can be collected at different stages of cancer through the model systems that have been developed, one can try to map this trajectory in terms of the genetic clonal signatures as to which signatures or which mutational signatures were acquired at what time point when a cancer was facing a particular bottleneck or was able to overcome a certain bottleneck. This relates to the idea of having fitness, that different genetic mutations basically alter the fitness of given cancer cells, that they can now adapt to the bottleneck that is being faced and eventually lead to cell growth and progression of the tumor. The overall goal to do this exercise is to try to identify, are there mutations, specific mutations against which there can be targeted therapies that can be developed, which can be then rendered onto the patients, which is what is called as precision medicine or personalized medicine. Now, the question that we ask in our group largely is complementary to this. So we don't focus on genetic heterogeneity per se, but rather focusing on another layer of heterogeneity, asking the question that if there are a bunch of cells which have the exact same genetic background, can they still have different phenotypes? So that is why we refer to it as non-genetic heterogeneity, non-mutational heterogeneity, because it is beyond the genetic aspects that is usually talked about in the context of cancer literature. So first of all, can isogenic cells, clonal cells, cells with the same genotype, can they have different phenotypes? And if yes, then how do we identify them and what is their potential role in the context of cancer? Now, this idea of non-genetic heterogeneity or phenotypic heterogeneity, whatever you would prefer to call it, is not something which is specific to cancer. In fact, this forms the central tenet of development of any given organism. So in the development of an organism, which usually starts with a single cell, and then the single cell can specialize, continue to specialize, and give rise to multiple different kinds of cells in the body, this question always comes up because during development, we don't really see genetic events happening in terms of mutations being selected or so on. It is the same genotype which is leading to different phenotypes or different set of behaviors, functional behaviors of a given cell. So this diagram here represents the cartoon of Waddington's landscape, which was proposed by the famous geneticist Waddington in the 1940s. And the questions that it tries to raise is, so let's say if you have this progenitor cell shown in the, as a blue ball, then how do we know which valley it will go to? So valley here represents some sort of differentiated cell type, the final phenotype that a cell will attain. And the landscape that it is following is somehow mapping the developmental trajectory of this individual cell or a set of cells. So if a cell is present at such a point in the landscape that it can end up into multiple valleys, then what decides which valley it goes to? And how do these decisions eventually impact the architecture, the function, and various other physiological aspects of organ development? Because in development, there has to be a recipe followed with a very specific spatial and temporal signature. However, in the context of cancer, it's largely believed that some of these boundary conditions are relaxed, leading to a more chaotic environment. But again, in development, this is tightly regulated overall. So that's why I just want to highlight the differences when I use these words genetic and non-genetic. Genetic is something which is heritable, which is largely irreversible once it's passed on from generation to generation, and it's deterministic in the sense. And phenotype of a daughter cell is, or sorry, the genotype of a daughter cell is going to be identical to the genotype of the parent cells. Non-genetic is sort of the exactly opposite. It's not necessarily heritable. It can be heritable because of some epigenetic marks and chromatin modifications, but it's not necessarily heritable. It is reversible. That's the biggest change. And stochastic, because again, as we were just talking about this individual cell, which has the possibility to give rise to any two of them. So which one it gives rise to is more like a flip of a coin. It's not encoded directly in the DNA per se. Now, this idea of non-genetic heterogeneity has been coming up sporadically in the context of cancer as well. There are reports across different cancers, say lung cancer, breast cancer, and also more recently melanoma, where this idea that the heterogeneity is not necessarily always at a genetic level, but the cells with same phenotype can also be behaving very differently when it comes to drug treatments, when it comes to metastasis, is something which is gaining quite some interest in the field. As you can see, these are all reports in leading reputed peer-reviewed international journals. Now, the other idea or the other representation one can think of this non-genetic heterogeneity is this very simple curve that many of you may have come across, the idea of IC50. So x-axis is the drug concentration and y-axis is the percentage of cells which is alive. So IC50 is typically the concentration of drug at which 50% of the cells die. And this is used as a benchmark for how sensitive a cell population is to a given drug or whatnot. But 50% cells die, but what happens to the remaining 50%? So there itself you have this heterogeneity, because you would think that these experiments which are done with cell lines are largely clonal populations, but still they are behaving very differently. Only 50% of them is dying, 50% of them is not dying. Of course, there can be this confounding factor that some cells are not taking up the drug to that extent. Sure, I agree with all those confounding factors. Now, this idea of, again, this phenotypic heterogeneity has been there in other contexts, taking a detour away from cancer. For instance, in the context of antibiotics, what people have identified over a period of 2 decades or so is that if you take a bacterial population and then expose it to increasing dosage or increasing time of a given antibiotic, what we see is that a large population of cells die, but a very small population of cells do remain. And if you take that small population of cells and reculture them, what you see is that you see the same graph again and again across generations. Now, if this were due to some genetic hard-coded, hardwired events, then what you would have seen is something more like an antibiotic resistant population shown by the blue line at the top, and not the same curve repeated again and again. So what has been identified in the field is that this is not due to genetic reasons. This is due to phenotypic differences because there are multiple states in a given population, and cells can stochastically switch back and forth among different states, which is what can give rise to this behavior. Similar reports have been seen in hepatitis C, more recently Mycobacterium tuberculosis, and even if one goes to the initial reports which were made by Otto Warburg, to which Warburg effect, one of the central tenet of cancer metabolism, is attributed. So the initial comments that were made were that there can be an irregular distribution of metabolic properties in cancer cells, in which a few cells would undergo glycolysis, whereas the others will not. So again, you have that heterogeneity idea right there present almost a century ago, but somehow it did not feature as well into the possible reasons why this exists and what can be the implications. So that is what we are trying to decode overall, because these non-heritable mechanisms of drug resistance have been observed across different biological systems. So the questions we are trying to answer is, where does this come from and what is its possible role when it comes to cancer treatment? So focusing on metastasis, which is a major focus of studies, is a process through which cancer cells move from one organ to another, which is largely through this fantastic set of freeways in our body called the blood vessels. So cancer cells in a primary tumor would get on these blood vessels, travel all over the body, take different exits at different parts of the body, and start to grow tumors there. Now, when I started looking into this problem, what was most intriguing to me was that the more than 80% of the cancers, they actually happen in those organs where the cells do not have this intrinsic property to start moving or invading. So if you think of the most common cancers you may have heard around you, breast cancer, prostate cancer, lung cancer, liver cancer, all of those organs are epithelial in nature. The cells are stuck in a specific 3D geometry very tightly to each other. They do not adhere, sorry, they do adhere very strongly. They do not necessarily invade or migrate on their own. So how do cancer cells gain this property? And then later, once they have reached a distant site, they again lose this property. So it's a reversible dynamic phenomena, not an irreversible phenomena per se. So during metastasis, cells are continuously changing and adapting to these different properties and their different environments around them. Because unlike a primary tumor, where the only objective of a cell is to continue to grow and develop at a much faster rate as compared to the normal cells, in metastasis, the objective of a cell is also constantly changing. It has to get out of the primary tumor. It has to be able to survive in circulation. It has to be able to settle down in a foreign organ, which is biochemically and biomechanically very, very different, and then finally be able to colonize that as well. So that's why we think of metastasis as a very dynamic process, and it's not at all surprising that metastasis is a highly inefficient process. Studies have shown that more than 99.9% of the cells which leave the primary tumor are not able to form metastasis. But still, metastasis remains the large reason for all cancer-related deaths. More than 90% of deaths are attributed to metastasis. Now, when it comes to, again, the genetic versus non-genetic discussion that we were having, there's a huge amount of money which has been spent on cancer genomics, but so far there is no unique mutational signature that has come out for metastasis. And The reason we rationalize this is that, again, as I was mentioning, that metastasis is a dynamic process, and the objective is also a dynamic function. It's changing as a function of time. So having a hardwired fixed solution may not necessarily be the best way to go about it, rather than having the flexibility or adaptability to be able to traverse that landscape where these changes can be made in some coordinated manner may perhaps be thought of as a hallmark for metastasis. So this overall idea comes from a whole systems approach, where again the central tenet is that whole is greater than sum of its parts. If you're given all the car parts which are not put together, that doesn't tell you how exactly a car moves. But when you put those parts together, there emerges a specific function which was not there in any of the constituent parts. So of course, molecular oncology over the past 6 to 7 decades has done a fantastic job in identifying all those different car parts, identifying all those different genes, proteins, non-coding RNAs, etc., which are contributing to this process of migration of a car or migration of a cancer cell. But what we have not done yet is put them together in a specific integrated framework to see how this function emerges overall. And that is where we hope to bring some of the contributions, where we can develop mathematical models for what is known in experimental biology, are addressing specific questions, and those models can hopefully generate some new hypotheses which can drive a whole new set of experiments leading to an overall better understanding of the system. And I'll try to give you a few examples of it during the talk. So again, just what exactly systems approach means. So here is the example of an engineered system. I compare it to a biological system. So this is some network of E. coli, which is one of the simplest organisms one can think of. And you see that the number of feedback loops which are present in E. coli are actually more than an order of magnitude more than the number of feedback loops which are present in the most sophisticated engineered systems that we can think of. Now, for these engineered systems, we have a design manual available. We know how to fix these systems when they go wrong. But for biological systems, they are evolved, they are not automated per se, so there's no design manual available. So how do we try to understand the property of this system and try to fix these systems is what systems approach try to do. So from the point of view of cancer, and of course, these circuits have been drawn by some of the leaders in the field and how these different molecules are interacting with one another. And that is where I would like to highlight the difference in the word understanding itself, as it may mean to a molecular oncologist versus someone who is looking at it from a more quantitative and predictive lens. So here is a network from one of the most cited reviews in the field of cancer called Hallmarks of Cancer. And this may give you a picture of how these different genes are interacting with one another. But when it comes to writing down mathematical models for this, there is no time scale information, length scale, no information of the strength of regulation, which link is direct, which is indirect, what is the nonlinearity of each interactions, what happens if there are two input nodes coming into, two input links coming into a same node. Are they additive? Are they super additive? Are they multiplicative? What exactly is going on? That quantitative information is just not available. So therefore, the clinicians are largely operating in some sort of a black box, and which can lead to various unpleasant surprises. For example, the anti-angiogenesis therapy, which is the therapy designed to stop tumor from growing its own blood vessels, which was given in early 2000s, had to be stopped. The clinical trials had to be stopped in between because patients receiving that some of that therapy were actually dying faster. And only later it was realized that once these drugs were given, what was happening is that metastasis was accelerating. So we cannot think of cancer as a static system which is not evolving, which is not responding to the perturbations which is given. Rather, we have to begin to think of how it may adapt, and can we outsmart it by identifying those adaptation mechanisms and then reducing the probability of letting it adapt. This is the last sentence of the most cited review in the field of cancer biology, which says that one day we imagine that cancer biology treatment will have a logical coherence that rivals that of chemistry or physics. These are those branches, again, where there is enough crosstalk between mathematical modeling development and experiments and so on. And again, this is the last sentence of the review, which I was just talking about, where again, the authors, which are the stalwarts in the field of cancer biology still think that those underlying organizing principles, the F equals to MA of cancer, has not been able to identify. So now again, coming to non-genetic heterogeneity, I'm sure many of you are familiar with these T as an E kind of plots, which have become very popular over time, where one can project the single cell RNA-seq data taken in multiple genes, huge high-dimensional data, and project them onto these 2 dimensions to try to see what different types of gene expression profiles, what different types of cells may be existing there. So the questions we ask is, where does this non-genetic heterogeneity come from? What is the role of this? And can we control this heterogeneity in a dynamic evolving system? So the approach that we take is a very iterative approach. We start with existing experimental data, construct biochemical networks for it, try to make mathematical models based on that, analyze those networks, and have those specific predictions from the network go back to either new experimental data gets collected as response to those predictions, or we validate our predictions against what already exists in the literature. So of course, this is a team effort. It cannot be done by a set of people alone. And we need expertise across different disciplines as well. So that's why what you see here is collaborators that we are fortunate to have, which includes oncologists, clinicians, mathematicians, physicists, chemists, engineers, all trying to understand what can we better understand the dynamics of this process and hopefully improve the therapies which exist in the clinic. So I've already introduced the idea of metastasis to you, and the overall idea that most of these cells are epithelial in nature. So the idea that exists in the field when we started looking into this is that some of these cells undergo what is called an epithelial to mesenchymal transition, where mesenchymal cells are the cells in the body which have the exact opposite properties as compared to the epithelial cells. They are able to migrate and invade, and they typically do not adhere to their neighbors as strongly as epithelial cells. So this is usually thought of as the first step in the metastatic cascade. And once it reaches the other organ, these cells are believed to undergo the exact opposite transition called MET, because now they no longer need to invade or migrate. They actually need to settle down and form a whole new tumor. So these cycles of EMT and MET, which continue to form secondary, tertiary, and so on metastasis. So in the EMT-MET paradigm, it was largely thought that cancer cells either don't move at all, which is epithelial phenotype, or they move as single cells, which is the mesenchymal phenotype. But there were reports showing that when cancer cells leave the primary tumor as a cluster, as a cohort, as a group, then they form much more metastasis than what they do as a single cell. So here in this experimental data, it was shown that clusters can form 50 times more tumors as compared to single cells. And it's not that these clusters are 50 cells large. These clusters are typically 5 to 8 cells large, but somehow they are able to form these tumors much more efficiently. So that is what we try to understand as to what exactly might be going on here. Sorry, I'm just trying to change the slide. So the question we started asking is that how do clusters fit into this binary paradigm of EMT, where the cells are thought of to believe as believed to be categorized into 2 of these phenotypes. Either they don't move at all, or they move as single cells. And these are just snapshots of reviews that have been written over a decade, where again, you would see is that the final behavior of a cell was always talked about as a single cell, and the collective migration, even if it was identified, was thought of as some intermediate step and not a stable phenotype in itself. So what we did is look through EMT literature and MET literature, try to identify what different molecules are known to regulate EMT or MET in different cancers, and how do they connect to each other? So I'm just showing you the first circuit that we made. Of course, as you'll see later, we have expanded this circuit. So here there are two transcription factors, snail and zeb, which are driving a more mesenchymal phenotype. So if the levels of these genes is high, the cells become mesenchymal. Then there are these two microRNAs, which inhibit a mesenchymal phenotype and drive a completely epithelial phenotype. And each arrow can be thought of as some quantitative relationship between input-output variables, which can be converted into the form of a mathematical equation. Trust me, this is the only slide with any equations, so please bear with me here. So what we can do is convert this network into a set of ordinary differential equations, or wherever required, partial differential equations, where you can track the change in time of each of these molecules and how they influence each other. So there are the usual terms of production, degradation, and the influence that they can have on one another through whatever parameters that are available in the literature. So what our model predicted was the following, that now if you take this network and the x-axis is some measure of external signal which is provided to a cell, be it a growth factor or other exogenous ones, which are trying to induce an EMT. And the y-axis can be thought of as a measure of EMT-ness. The solid lines are the stable states and the dotted lines are unstable states. So what this model tells us is that, of course, you have an epithelial phenotype and a mesenchymal phenotype as reported in the literature, but you can also have, in addition, a third phenotype, which is more somewhere in between, the hybrid epithelial mesenchymal kind of phenotype. And this particular phenotype, as we have shown via experiments later, can actually have the property of collective migration because it has the epithelial property of adhesion and the mesenchymal property of migration. So it can somehow have the best of both worlds and exhibit that particular phenotype. So that's one prediction, that EMT is not binary. You can actually have a third state where cells can stably stay. It's not just an intermediate state. The other prediction was that now if you look at this graph vertically upwards, so you have a region in which the only phenotype that is available is epithelial. You have a region in which the only phenotype that is available is mesenchymal, but you also have regions in which there is more than one phenotype available. What that tells us is that it is possible that once you take a genetically identical population, you still see cells in three completely different phenotypes, and also they can interconvert because there is enough noise which is present in the system. So these were the three different predictions that we had, and then we established experimental collaborations to actually test these predictions. So the first one saying that cells can actually stably exist in hybrid epithelial mesenchymal phenotype. This was shown in lung cancer cells, so in this image, the green is staining for an epithelial marker, the red is staining for a mesenchymal marker. And what you see is that single cells actually co-express both red and green markers, and they don't only do it at one given time point, but they continue to do that over multiple passages seen in the lab over 2 months, suggesting that it's not a snapshot, it can actually be a phenotype in itself. We also looked at other cell lines which are widely used by various different groups, and observed that you can actually have cells in three different phenotypes when you do flow cytometry: epithelial, E-cadherin, which is an epithelial marker, positive, vimentin negative, vimentin positive, E-cadherin negative, the second and the fourth quadrant shown here, and the first quadrant in this graph, which is the double positive, where cells co-express these markers. And then there was other set of experiments where people segregated these three different populations and then just cultured them independently. No treatment was given, just they were segregated from one another. And what you see this experiment shows is that the cells actually switched. Over a period of 2 weeks, you see that epithelial and mesenchymal were largely able to maintain themselves, but the hybrid one actually switched largely. So now this drives the whole next set of questions is, can we explain this population level dynamics feature? So far, we only looked at intracellular signaling. There was no information on population dynamics. So again, the question we asked here was that what happens to the fate of a cell when it divides? When an epithelial cell divides, does it always give rise to two epithelial cells? Or does it never give rise to two epithelial cell? Or again, is that noisy? Is that a flip of the coin? Is that probabilistic, stochastic? So under what assumptions will we be able to reproduce whatever that experiment saw? So we were able to actually match that experimental data that I just showed you under the approximation, under the assumption that whenever a cell is dividing, there is 10 to 15% variability in distribution of the molecules. So not every daughter cell gets n by 2 molecules, but a daughter cell may get anywhere between n by 2 plus 10% and n by 2 minus 10% in that distribution, which is a fair approximate of the distribution of noise as has been experimentally seen. We were able to explain that. And we were also able to say that because the percentage of the cells in different phenotypes is a dynamic variable, actually one cannot necessarily say that the so-called cell line is always epithelial or always mesenchymal, because these dynamic processes are constantly happening. So we can say it's largely epithelial or largely mesenchymal, and that's just the distribution that you see in some EMT measures, which I'll come to in a minute. But again, you see that they span, many of these cell lines span a large spectrum. So that brings us to the idea that, yes, there are multiple states, as going back to the Waddington landscape I talked about earlier. And cells can switch back and forth because of noise, because of cell division, etc., which can often lead to scenarios that even if you start with only one subpopulation, there is some probability that it can give rise to others. So there is this heterogeneity which is always there. Now, this is where I moved to ISC. This question we started asking is, well, what is more important for cancer? Is EMT more important, or the ability to regulate EMT in a dynamic fashion more important? So we started asking questions. Can we block both EMT and MET together? Because what is going on in the clinic at this moment is that there are different inhibitors which are blocking EMT, but they may actually be promoting MET. So these drugs, which we are thinking of blocking metastasis, may actually be helping metastasis. So are there ways to block both of them together is the question we started asking. So this is where we looked at what others modeling groups had done in the context of EMT. So once we started the work in 2013, there were various other groups which started modeling EMT networks of different sizes using different approximations and so on. But the general idea that was there consistently across all these different models was that EMT is not binary. Cells can actually stay stably in one or more hybrid phenotypes. Now, as some of you are more familiar with the modeling, the mechanism-based modeling idea, what happens as the network size grows, you need more and more parameters. And those parameters, kinetic parameters, biochemical parameters from experimental data. And those parameters are rarely available. So what people do usually is something called as a Boolean modeling, where what one assumes is that if A activates B, if A is 1, then B is 1. So it's a very discrete way of looking at the dynamics. So it gives a very good qualitative idea as to what this network is likely to do. But when it comes to quantitative detail, then this particular method is not necessarily the best one. So what we did here is we compared the two different methods, one in which you don't care about parameters at all. Say that, sure, they don't exist, Boolean model. And the other in which we said that, yes, parameters are important, but what we are going to do is, because we cannot determine the parameter in individual cells with absolute precision, we'll take the range of these parameters. So let's say if the half-life of ZEB protein is anywhere between 6 hours to 14 hours. I'm just making this up hypothetically. So then we'll consider a population of cells, and each of these cells can now have the half-life of ZEB picked up from a normal distribution between 6 hours and 14 hours. So we took an ensemble of such scenarios. So in this case, you are saying that parameters are important, but we are taking a large ensemble. Therefore, probably, again, they might not matter. Who knows? So that's the parameter agnostic approach that we're talking about. So the question we wanted to answer here is that is just the topology of a network sufficient to convey some information about its behavior without depending on parameters? And this work was led by my PhD student Kishore and two summer interns who spent the last summer in the lab. So we looked at different EMT networks of all different sizes, starting from 4-node networks all the way to 30-node networks. I'll just present a summary of the results here. And what we found was that indeed, at least for smaller networks, the distributions that we got in terms of steady states and phenotypes that these networks can give rise to depended largely on topology and not necessarily on the parameters per se. And then we asked this question that, can we figure out a specific feature of these topologies which will tell us whether this network will enable plasticity, plasticity being the ability to switch back and forth, or it will inhibit plasticity such that it will reduce adaptability and sort of force cells to stay only in one phenotype, not giving them the option to switch to another as and when required. So what we found was that the larger the number of positive feedback loops in a given topology, in a given network, the higher the plasticity score. So again, suggesting that every cell may have very, very different parameters from one another, but if it has more feedback loops which are operative, then it is likely to be more plastic. That was the prediction. And therefore, breaking these feedback loops can actually reduce plasticity. So there was a paper in Nature Communications almost 2 years ago, which actually already did this experiment, which we predicted. So they looked at this feedback loop where ZEB and MIR200 inhibit each other. So this is the loop that I've been talking about in the first half of the talk. So overall, it's a positive feedback loop because ZEB is inhibiting its inhibitor. So ZEB is overall activating itself. So what you see is that using CRISPR, they were able to break this feedback loop. And once this feedback loop was broken in this mutant cell lines, metastasis actually went down. This is the last figure of the paper. They don't offer a mechanistic explanation of why this happens. But again, one can map this onto the predictions of our model and say that the possible reasons for this, why this may have happened, is because A, the cells have lost the ability to switch. B, if you have cells of two different kinds, they may be cooperating with each other in all different manners. And of course, there can be a combination of those two factors. Now, these feedback loops doesn't only exist in EMT. We have looked at feedback loops in other contexts. So this is work, ongoing work in collaboration with Professor Rangarajan's group at MRDG here at IISc. So they had figured out another double negative feedback loop, which helps cancer cells adapt to once they are deprived from the matrix, because when they are leaving the primary tumor, in the primary tumor, they are getting all the signals from the extracellular matrix. But once they are deprived, many cells actually die because they are so-called addicted to biochemical signals and biomechanical signals receiving from the matrix. So again, there's a double negative feedback loop which operates there. We did some simulation, some initial simulations. We see that this network also has the ability to give rise to more than one phenotype. And we then suggested them the same experiment which I talked to you earlier about the EMT context, that why don't you start with one subpopulation and then see whether it gives rise to the other. This is a specific property of multi-stable systems wherever there is more than one phenotype available to the cell. And this is what they did. They sorted the cells into high and low based on the reporter system that they had developed earlier. And these are supposed to be two different states as shown by a simulation. So what you see is, as a function of time, the red population does give rise to green and the green does give rise to red. The rate of switching may be different. That's a separate question which we are still looking into. Another set of undergraduates who worked together wanted to look at this idea of feedback loops and plasticity in a completely different disease I had never studied earlier, called non-alcoholic fatty liver disease, which is actually the most common chronic liver disease. And again, there is no established guidelines for treatment as of 2019. What happens in this disease is that in some of the liver cells, which are hepatocytes, fat begins to get accumulated. So these cells begin to show some properties which are usually associated with an adipocyte, which is what the fat cells are. So they identified a particular network, again, going to the literature, just as we identified an EMT network, they identified an NAFLD network, where again you have these hepatocyte fit markers, HNF1 alpha, HNF4 alpha, which maintain a healthy liver, a hepatocyte state. And then you have two other so-called master regulators of an adipocyte-like state. And again, what you see is they inhibit each other. The same trend seen in EMT, the same trend seen in matrix deprivation, and the same trend seen here. So this seems like a very universal principle that when there are networks of this particular topology, it usually leads to two completely different phenotypes, and it has the possibility that these phenotypes can switch back and forth, which is what they are showing here using stochastic simulations, that actually cells can go back and forth. Now, this was all the mechanism-based aspect. This is where we started getting interested into, can we now look into the gene expression data and try to make some sense of it from the perspective of what we have been saying. So EMT is clearly a highly nonlinear process and multidimensional process because various different properties of the cells change. Cell adhesion, basement membrane remodeling, polarity, motility, etc., etc. And although these are being shown as independent axes here, they are actually not independent. They influence one another. So what may be happening is that when EMT is induced by one transcription factor versus other transcription factor, so different transcription factors may be pushing different cells to different extents on these different axes. So is there any universal signature of EMT which can be identified in the gene expression landscape? So this is just shown by a schematic of principal component analysis, saying that EMT induced by one transcription factor may be very, very different from that induced by another one. So we started asking this question, that what methods are there in the literature which quantify the extent of EMT in an omics data? And what different algorithms did they use? What different gene lists do they use? And is there any consistency in those EMT scoring metrics? This work was led by Priyanka in the group. So we looked at what different signatures are out there. We had developed one of them 3 years ago, the MLR method, shown in panel B. And then there are two other methods, at least these two other methods, which exist in the literature for transcriptomic EMT categorization. And one of them is shown in panel A, scores epithelial cells with a higher score, and mesenchymal samples have a lower score. The other two methods score mesenchymal as higher and epithelial as lower. Each of these use different number of genes. The first one uses 76, the second one that we developed uses 23, and the third one uses anywhere between 200 to 400 genes. You will also see the difference in their scaling. MLR and KS scale them any given sample on 0 to 2 or -1 to +1. But for 76GS, there is no very clearly defined scaling there. So for instance, for MLR, what we would say is that if a score is close to 0, it is much, much more epithelial. If a score is close to 2, it's much more mesenchymal. And it's a continuously increasing score on 0 to 2 or -1 to 1. So the first thing we did is to look at a large number of cancer cell lines and then see if we calculate their scores using each of these methods and then do a pairwise correlation, is there any consistency? So because 76GS scores epithelial as the higher one would expect that scores by 76GS method correlate negatively with MLR and KS, whereas MLR and KS should correlate positively with each other. So that's what we saw with a high correlation coefficient, statistically significant, this trend exists. So, okay, there is some hope that in terms of quantifying EMT. The next question we asked is, now let's look at these 900 cancer cell lines and break them based on their primary site of origin and ask the question, are there some cancers which show more heterogeneity in EMT as compared to others? So what you see in panels A through C is we have broken these 900 plus cancer cell lines into primary tumor cases, site of origin. And then for say 50 cell lines in breast cancer, we calculated the mean and we calculated the variance. And that is what is plotted on the X and Y axis respectively. Again, what we see is a very common trend that all three methods seem to identify a common set of cancers which are much more heterogeneous as compared to others. So the ones which are highlighted, and you would see the breast cancer, stomach cancer, bile duct cancer, and lung non-small cell lung cancer are the ones which are actually more heterogeneous in terms of their EMT. And again, what we observed was that cancers in which there were more hybrid samples were also the ones which are more heterogeneous. Because think of hybrid as the one which is somewhere in between, as shown in the initial graph there. So it has the maximum ability to go either left or right. And therefore, it is likely to have a higher ability to give rise to more heterogeneity as compared to the ones which are stuck on either end of the extreme. So the next question we asked is, sure, there are some cancers where the heterogeneity is more, but does that mean they are more aggressive? So we looked at this example of what is called inflammatory breast cancer, which is a highly aggressive form of breast cancer. It constitutes only 2 to 4% of all breast cancer cases, but 10% of the mortality of all breast cancer cases. It is present in all subtypes, and it does not show any mass or lump which can be detected by mammography. So the detection is also very hard. What has been shown is that this particular cancer moves largely via collective cell migration. If you delete the cell-cell adhesion molecule, which is usually thought to drive metastasis, in this way it happens the exact opposite, and metastasis actually goes down because they are moving as a group, and the moment you sort of segregate them from each other, separate them from each other, united they stand, divided they fall, they don't metastasize. They lose their ability to metastasize. So what we did here is, again, let's go back into the literature. Let's see, have people come up with gene lists which can be used to identify IBC versus non-IBC. So IBC versus non-IBC categorization in these papers have been done by a pathologist, and then they have developed different methods to come up with this. 4 different papers, 4 different gene lists, no common gene at all. So is IBC so heterogeneous that one cannot come up with any common gene signature, or is there something else that is going on? Here is the question we started with. There was only one exception. 2 of these gene lists shared one gene, but that's about it. So then we asked the question, now let's calculate their EMT scores and see if any trend shows up there. So in each, we found 3 publicly available data sets in which we had this information on which is IBC and which is non-IBC. And we calculated the mean using 3 methods and the variance. What we see is one trend stands out very clearly. So 3 data sets across 3 methods, 9 total possibilities. Out of 9, in 8 scenarios, we see that the IBC is more heterogeneous than non-IBC. There's no trend in the mean. Means vary all over the place, but the variance is very clear. So suggesting that this high heterogeneity in EMT itself can be a hallmark of this highly aggressive disease. And we have shown that this correlates well with the single sample GACS score, and also help improve the categorization of samples into IBC and non-IBC. We are hopeful that we can be able to test this hypothesis when it comes to single cell RNA-seq data. Currently, there is no publicly available single cell RNA-seq data in terms of IBC, but we are hoping once the data becomes available, we can test this hypothesis even more clearly. And on a more mechanistic reason, can we try to answer these questions as to why high heterogeneity will lead to a more aggressive tumors per se? So again, I'll just conclude here. As I mentioned that the existing framework had been that the hybrid state is transient, and the more the EMT, the more aggressive the cancer. What we proposed is that actually hybrid state can be stable and maybe perhaps even more aggressive than a complete EMT. And this idea is becoming more and more popular in the literature. These are just snapshots of some reviews that have been written after we published some of our work. So EMT connects with various different properties. I only talked about migration and invasion, but it also leads to chemoresistance. It leads to resistance to cell death and eventually affecting patient survival. And we are looking at some of these aspects together with the different collaborators that I mentioned earlier. And the central question that we try to answer, again, coming back to metastasis as a dynamic process, is the following. So this is a very simple math question that So if you say that a box has 6 white balls, 3 red balls, and 3 blue balls, in how many ways can one pick 1 white, 1 red, and 1 blue ball? So each of them is an independent experiment. There are 6 ways to pick up a white ball, 3 ways to pick up a red ball, and 3 ways to pick up a blue ball. And since these are independent, we multiply the possibilities. So there are 54 ways to do that. Now, how does that connect to EMT and metastasis? So people have identified that there are 6 or more EMT states. There are 3 or more so-called stemness states. There are 3 or more so-called metabolic states. And you can continue identifying more and more states along each of these supposedly independent axes. But when it comes to a cell, how is it coordinating all these different properties together? Is it just by chance, or is the network between these molecules regulating each of these axes, is the topology of the network somehow constrained to give rise to some states which are more fit as compared to others? So can we identify those states? Can we look at the topology? And finally, can we validate it with the now available single cell RNA-seq kind of data, where you can actually map this data on different axes comprised of different components of these different genes. So I'd just like to end here. The questions that we started with is, where does this non-genetic heterogeneity come from? What is the role of this heterogeneity? And can we quantify this heterogeneity as a hallmark and control this in a dynamic evolving system? So we continue to work on these open questions. Some answers that we seem to be getting closer to is that mechanisms leading to this heterogeneity can be multistability, the ability of a network to give rise to multiple phenotypes, possible noise in gene expression, cell division, etc., etc. Of course, there are extrinsic factors that the matrix stiffness can be different. And of course, there is genetic heterogeneity. The genetic background and epigenetic background is also playing a role. In terms of what is the possible role, I didn't get to talk about this work, but some of the work we have looked at suggests, similar to what has been seen in bacterial cell cultures again, is that there is a reason for having this heterogeneity, because it may lead to the population as a whole being able to survive various different bottlenecks. With that, I would like to thank you all for your attention, and I would like to thank my group members who have been working hard to address some of these questions. And I'd be happy to take any questions. Thank you so much.
Thank you so much, Mohit, for this wonderful, wonderful talk. And I think I cannot think of a better talk in terms of computational modeling of the molecular principles of metastasis. I think I will open up for questions from the participants, but to begin the question answer round, probably I would take the privilege to ask the first question, which may not be directly related to the talk. So my question is, as you mentioned, and many of us probably know, that the 90% of the cancer deaths are attributable to metastasis. My question is, how much EMT is responsible for metastasis? Is there a way to quantify that? In which ways, right? Because I can think of other mechanisms apart from EMT. They can potentially exist that can give rise to metastasis of cancer epithelial cells. So can you think of other mechanisms, and is there a way to somehow rank order them in terms of their implications, right, or importance in the process happening? So that's my question. Yeah.
Sure. No, I think that's a very important question, Debarka. Thanks for starting with that, because that goes back to some of the ideas we was talking about, that metastasis is a high-dimensional, nonlinear, dynamic process. So cells need different properties at different instances to be able to finally get through that bottleneck. And it being such a highly inefficient process that 99.9% of the cells are not able to finally establish metastasis. So we know, compared to what we know about primary tumor, we know very little about the dynamics of metastasis. There are various possible reasons. The experimental model systems are not perhaps as sophisticated as we would want them to be. So for instance, whether metastasis is an active process or a passive process, I'm not sure there's much clarity on that. So the EMT would lie to much more on the active side of the argument, that cancer cells actually go and crawl and pierce the blood vessels by breaking that endothelial barrier and enter. But there have been reports saying that metastasis can be a so-called a passive process, where the blood vessels which exist in primary tumor are leaky in nature. They don't have an intact vasculature, and that just happens to engulf the cancer cells which are present there. So now, will they show in EMT markers? Who knows? There's no clarity on that. What has been seen in that in circulating tumor cell, the liquid biopsy scenarios, that there is huge heterogeneity along the EMT spectrum. So you see epithelial ones as well as the mesenchymal ones, and that again also was very clearly shown in the work that you had led together with Naveen, that there is heterogeneity. So what is the functional contribution of each of these subsets still remains to be identified. So EMT plays an important role, and maybe interconnected with various different other axes that you mentioned. So now whether we call that axis EMT or we give it a different name, I'm totally agnostic to that as far as we understand the molecular principles.
Absolutely right. And I think, as you mentioned, that in our current work where we collaborated, we saw there is a huge heterogeneity in the CTCs, circulating tumor cells, and it does not necessarily tell that everything is in transition. Thanks for the answer. So, anybody else wants to ask a question?
Hi Mohit, this is Gaurav this side. So I have a very small question because I have another meeting as well. I have to rush. So I mean, it's very nice in your beginning of your slides. You showed like the you have shown the similarities between EMT transitions as well as self-hit commitment from the ES cells to the differentiated states. So my question is, for example, is there something, for example, in the ES cells, we know like there are some factors like Yamanaka factors, which are basically NANOX, SOX2, OX4, KLF4, which are basically potentially required. Equilibrium between all these four factors are essential for the stem cells to maintain their pluripotency. So in the same case of E to M transitions, do we know what are the core set of genes which are basically responsible for maintaining the epithelial state? And in all, I mean, since you have talked about there is a huge heterogeneity whenever there is in the hybrid state, we have so many different cell types with different transcriptomes. So is it somehow linked with like, for example, there are 5 different core genes which are maintaining the epithelial state. And each of these 5 molecules are in highly tight equilibrium so that the cells can retain their identity of being epithelial. And the heterogeneity could be the loss or the combinatorial loss of any of these 5. Is there something like this in the case of EMT transitions, like what we know for the stem cell differentiation models?
Yeah, thanks for the question, Gaurav. I think that's it's a very open question at this moment, is what I would say. So in the ES cells, as you rightly pointed out, there are these Yamanaka factors which have been clearly shown to be able to maintain that state. So what we are doing currently is actually looking at these interconnections. So we are going through some of the iPSC data. And in iPSC, the EMT and MET have been reported. So in that reprogramming experience, EMT, MET play a role. We're still trying to quantify that. But coming to your question in terms of, is there a core set of genes? So that is what we are trying to get to through this comparison of different metrics. So now that we know that all these metrics are able to largely capture what is going on, now we are looking into what genes are common in these different metrics. And then also looking at publicly available data sets where EMT has been induced by 10 different methods, and then seeing is there a common gene that shows up. So having said that, one debate which still is an open one is that, so EMT is not only a gene expression change. EMT involves changes in cell shape, etc. So there are various groups who don't care about gene expression, who are caring about the changes in cell shape, and they are defining EMT that way. There is a whole new set of people doing the gene expression way. So is there any consistency here is something which we are still not very clear about.
Okay, so sorry, since I'm running. So I have another follow-up question on that. Like whenever we talk about EMT, so this is a transition, and you have beautifully shown like there is a hybrid state in between. So my follow-up question is: do you also see a variability in the population of the mesenchymal cells? I mean, you see a lot of heterogeneity within the transition, which I appreciate from your data. But whenever the cells attain the mesenchymal state, there is also a variability in contrast to the epithelial state in terms of the transcriptomic variability within the isogenic population, because these are all the same cell types. So is there is a transcriptomic noise among the population of the mesenchymal cells?
Yes, there is. I would say it's slightly less than what is there in hybrid, but it's there. It's not a deterministic completely setup there.
And how it is compared to the epithelial state? When we compare it to the epithelial state, like the in an isogenic population of the epithelial cells, how the cell-to-cell transcriptional noise, or you can say any term, I mean, transcriptional heterogeneity, how it is related to the mesenchymal state?
So I'm aware of only one paper that appeared recently, which has single cell RNA-seq data of epithelial cells with and without being induced to undergo EMT. So
okay,
that is the first data set I know of where we can even begin to answer this question. Because I mean, we can't compare carcinoma cells with sarcoma cells, for example, because those are developmentally so different. So they are epithelial and mesenchymal, but comparing them, at least in my opinion, would be like comparing apples and oranges. So we will. That's a very important question. We look into it in this data set that has just appeared.
And sorry, and I here end my question, so I can also contact you later. So my last thing is, I mean, people always talk about like the unidirectional flow of EMT, and you have said about this hybrid state. So is this hybrid state is reversible?
Yes.
Within the hybrid state, or maybe you have told I missed that. So
Yes, it is reversible in both directions. So this can go to epithelial as well as this can go to mesenchymal. So it
okay,
can.
so it can go to either way. Okay.
Yes.
Thank you, Mohit. My questions are addressed. Thank you.
Thank you. Love to chat more. Yeah. Thanks.
So is there anybody who wants to ask any other question or? Okay. All right. If there is no other question, I think Mohit also informed me that he had a hard stop. So I'd rather let Mohit go. And thanks again, Mohit, for the wonderful talk and accepting the invitation for starting our webinar series. And it's a wonderful talk. And thank you very much. I'll get back to you.
Thank you so much, Tabarka. This was excellent. I'm looking forward to more interactions. Thanks once again.
Yes. Thank you.
Thank you.